on H7N9 influenza virus. I'm going to discuss today in general about H7N9 viruses and introduce influenza virus in general to give an understanding of how the H7N9 outbreak has gotten to be very concerning. So the learning objectives for today are that after today you'll be able to describe the characteristics used to assess the pandemic potential of influenza viruses. You'll be able to recognize the current impact and potential risks associated with H7N9, and you'll be able to describe mechanisms for worker protection and exposure prevention to avian influenza virus. So just to start with a brief overview of influenza, there are many different kinds of influenza viruses, and typically in the public health arena we're talking about two of those types, types A and B. Type A influenza viruses do include things like avian influenza and swine influenza, which we'll talk about in more detail, because they can go to multiple hosts as well, those animals and humans. Type B influenza, which also can cause pandemics, is a human-based flu virus only. And so we won't talk anymore in specifics about the rest of the influenza viruses, because the type A viruses are the ones that have the ability to exchange their genomic material, move between species, and H7N9 is a type A influenza virus. Typically and commonly, we refer to influenza viruses by their common HN designations. The H stands for the hemagglutinin. There are 18 different kinds of hemagglutinin, and the hemagglutinin, is one of the glycoproteins on the outside part of the influenza virus. This slide shows you what the influenza virus looks like, and the hemagglutinin would be the blue spikes on the outside. It's a protein that will bind to our cells, and we're going to talk about the receptor binding in a few minutes because it's really important for host determination, and it's really important when we talk about H7N9. Neuraminidase is the N. Neuraminidase is also on the outside of the influenza virus, and it's shown in this picture as pink. And this is a glycoprotein, but it also produces an enzyme which helps to break the binding of the virus to this host cell. This is really important for infection and also release of virus particles. Influenza virus is a single-stranded RNA virus. It has an envelope. The envelope is that circular portion of the virus you see where the hemagglutinin and the neuraminidase are located, and it has a segmented genome. And as we talk in the webinar today, the segmented genome is really important because it allows the influenza virus to share single genes between different viruses that can then all be packaged and create a new unique influenza virus. This has resulted in previous pandemics such as the H7N9 pandemic in 2009, where 4 different flu viruses were able to combine their DNA and create a new version. So we'll talk about that more later. And like I mentioned, pandemics, mainly type A and B. So here are all of the different genes in the influenza virus. The main ones that we're going to talk about today are the hemagglutinin and neuraminidase, which are shown at the top, and then also PB2. I'm not going to speak about it too much, but it is important in that there are a lot of mutations in this gene that are showing and conferring the virus to be able to replicate in mammals. And so if it's an avian type virus and there are specific mutations in this gene, those mutations are allowing the virus to adapt better to mammals. And this is being shown a lot in a ferret research model and confirmed in sequencing of a lot of the clinical isolates that we're seeing coming out of China. Influenza viruses, on the right-hand side of the slide, you'll see how they're typically designated. So in the common conversation and also on the news, a lot of times we just hear H7N9, which is the hemagglutinin neuraminidase components of that virus. This is important because they are on the outside of the envelope of the virus. They are the protein antigens that our bodies recognize and build an immunity to. However, that's not exactly how the virus is named. The virus is named with the information that you can see here on the slide. And so it's the A, which is the type of virus. Duck, it stands for where it was isolated from, which species. Alberta is the location. and then the year and a few other things. And so this, you'll see this notation specified a lot, and each virus isolate will have a different specific name. And so when you're looking at H7N9, you have many different specific strains of the virus. And so when we talk about host cell binding, here's a picture of influenza virus binding to a host cell. And so again, you have the hemagglutinin and neuraminidase on the outside of the cell of the viral envelope, and those little blue chains are on the cell membrane of our cells. And those chains are sialo-oligosaccharides. And so the influenza virus is going to bind specifically to the sialic acid receptor on those oligosaccharides. And it can do this in different ways. There are preferential binding sites depending on the type of influenza virus that this virus is. And so, you know, many of you are aware flu viruses can be in humans, they can be in birds, and they can be in swine population. And we're going to talk about the mixing of those in a minute. But the binding is really crucial and helps keep these viruses in separate organisms. And so if you have an influenza virus and it's a avian-only influenza virus, it will preferentially bind to the receptors that are at 2,3 linkage with the galactose. So basically, in these oligosaccharides, they have a galactose sugar in them, and so the virus is going to bind preferentially to ones that have a 2,3 binding. The human receptor is a 2,6, and so they don't tend to mix well. You have to, an influenza virus that binds to the 2,3 cannot bind to the 2,6. Swine actually have both of these kinds of binding receptors, and that's why they're typically known as a mixing vessel. And then poultry also can have both of these binding. And so this is why those organisms can actually sometimes have multiple flu infections, which allows them to share their genetic material. And so, as I was speaking a little bit about the life cycle of avian influenza viruses, avian influenza is naturally found in wild waterfowl. Type A influenza viruses are known to exist there, and for the most part, these animals are what are called reservoirs. The animal can be infected and show no symptoms of disease, and they're perfectly healthy, and there's no evidence that it's even in that population. But the wild waterfowl can have contact with domestic waterfowl, like the birds we see in our parks and ponds, and those animals may also come into contact with poultry. The overlap with these animals tends to be found in avian farms or avian slaughterhouses. And so you may have animals that are naturally infected with these viruses and harboring them, and then if you have a sick animal, or because they're excreting it out of respiratory secretions or feces, then you may have a mixing, and because the virus genome is segmented, different viruses can exchange different pieces of DNA or different pieces of RNA, excuse me, and so then you get a new virus. Or alternatively, the viruses, because they do mutate very fast, may mutate slightly and cause infection in a different bird species, or we go from low pathogenicity to high pathogenicity, but I'll talk about in a second. And so because the poultry have the binding potential for both avian-specific and the human-specific influenza viruses, and humans tend to come into more contact with poultry animals than with wild waterfowl, this is where you know, the human exposure can come into place. And so when we talk about H7N9, we'll talk about it repeatedly throughout the webinar today. The best prevention is to avoid contact with avian species, so that way you don't acquire infection. And then at the bottom right-hand corner of the slide, you can see the picture of the pig. And so, like I mentioned before, these are also a mixing vessel because they maintain the ability to be infected with both avian-specific and human-specific influenza viruses due to the binding potential on their cells. And so they then, if in contact with humans or with birds, can mix and then spread a different and new strain of influenza virus. And so this is how the life cycle occurs. And over time, these viruses are always mutating anyways and do slightly change. And so breaking down avian influenza viruses more specifically, you may have heard of two categories, the highly pathogenic avian influenza and the low pathogenicity avian influenza viruses. I've highlighted the highly pathogenic avian influenza viruses in red because in addition to causing very severe disease in birds, it is also categorized by the Centers for Disease Control and Prevention and the United States Department of Agriculture as a select biological agent, which means it's highly regulated. If it's identified in a laboratory setting, it must be reported to either the CDC or the USDA. And then if you don't have permission to maintain it, you must destroy it. And they've also now, in China, are requiring proof that the viruses that are isolated are of the low pathogenicity, or if not, in order to ship them to the United States, they must be declared as high pathogenic, even if, because if you don't know, they want you to overclassify them. The difference in terms of symptoms for the bird population is that the highly pathogenic avian influenza viruses are very severe in the birds. You will have a lot of sick animals, a lot of mortality, and it will be very evident in the population that there is an illness there. Whereas with the low pathogenicity avian influenza, it's not evident that there is illness, and again, these are just reservoir, and for the most part, they're just passing them along, and the virus is just moving between the different animals. What's really concerning with the current outbreak in China is that, this outbreak has been going on since 2013, but a lot of the H7N9 viruses are starting to acquire mutations that are changing its category from the low pathogenicity to the highly pathogenic versions. This is more of a concern in terms of the avian population than for humans, and we'll talk about that in a little bit. But it's also a potential economic issue. So avian influenza is listed as a select agent, not so much because it can cause a lot of infection in humans, although if it did cause infection in human and caused a pandemic, humans do not have immunity to these viruses. And so, because they're avian-specific only viruses, our bodies do not recognize the hemagglutinin and neuraminidase on the outside of that envelope. But also, it can be a severe economic issue because in order to contain outbreaks of these sorts, they typically tend to cull the poultry population and shut down those farms in order to eradicate the highly pathogenic strains. And so this can have an effect on the chicken that comes to market and the food supply for the population. And so this also is a good example to talk about the difference that we hear a lot with influenza viruses in terms of antigenic shift versus antigenic drift. And so in general, this is a slide from CDC, and it talks about how the H7N9 virus in China originally developed in 2013. And so, as I described before, the virus, as you can see in the center, has components from 3 different types of avian influenza viruses. One from an H7N3 virus coming from domestic ducks, one from an H7N9 virus from wild birds, that would be the neuraminidase component, and then the rest of the genome comes from domestic poultry. And so because these animals are in live bird or poultry markets and they're in close contact, they can share these viruses. And so with that, we saw an example of the antigenic shift, which is a creation of a whole new virus that hadn't been previously seen. Since 2013, there have been repeated outbreaks of H7N9 in general in China. The most recent outbreak in 2015 I'll speak more about in a moment, but since the 2013 outbreak, we're really seeing examples of antigenic drift, where the viruses are acquiring additional mutations in each of the genes in the genome that are conferring additional abilities. And so in some cases, they're acquiring mutations for the ability to infect mammals. In some cases, they're acquiring the mutation to spread by the respiratory route between mammals. although those are few, etc. The mutations that they are also seeing, though, include the drug-resistant mutations, which are common in H7N9 and just in general to avian influenza viruses, to the NA inhibitors. So I'll talk more about that in a few minutes. So the current situation in, with the H7N9 right now is that there have been 5 total epidemics in China since 2013. They're seeing these epidemics spread to larger geographical locations, and the current one since 2000 and the one currently in 2017 is the most concerning because it's spanning quite a wide range of geographical locations, and the viruses have gone from the low pathogenicity to the high pathogenicity. There have been a total of over 1,500 infections as of December 1st. The concern, though, with these infections is that they're not typical flu-like symptoms. A lot of folks have been getting pneumonia, and because of the lack of immunity, about 40% of people with infection are dying. And so typically when you see avian influenza infection, you do need antiviral treatment as soon as possible to help lessen the symptoms of the infection and the severity. And so the CDC, and we're going to talk in a few slides about how they've determined this, has determined one of the viral strains that they've isolated during this pandemic to be of high concern in terms of the overall pandemic potential, according to the risk assessment tool. I would like to point out that the risk assessment tool looked at one of the low pathogenicity avian influenza strains. It did not look at the high pathogenicity strains. And so the data that I show you in a few slides will be of an LPAI, and so I would imagine that the concern would be greater for one of the new high pathogenicity strains. The most important take-home message, though, about the current situation is that it is very rare for people to become infected with this virus. You have to have contact with birds. There is some transmission from person to person, but it is not sustained. They are seeing that they call them clusters from each epidemic, that there's more clusters that are coming for 1 to 2 to maybe 3 people getting sick from somebody who had contact with birds, but those are typically people in their immediate family or in their very, very close contacts, or maybe providing them a lot of care. These are also different strains than we have in the United States, and I'll speak a little bit about one of the outbreaks of H7N9 in the United States. But if you hear about it on the news and you hear H7N9, a lot of times they're not differentiating the strains that we see in China versus the strains we have here naturally in the United States. They are not the same. That is why the United States Department of Agriculture has very strict requirements about importing of any kind of bird products or strains of these materials into the United States. So this slide is not easy to read at all, but I want to just point out that there are many different H7N9 viruses. So this is what's called a phylogenetic tree. It measures relationships of different viruses based on genetic sequence. And so this is looking at viruses isolated in the outbreak since 2016, and they're color-coded. And the candidate vaccine strain is in the lower left of the phylogenetic tree. You can see it in red all the way down at the bottom. Each of the outbreaks has strains that are clustered in different groups, but the most important thing to notice is that in the 2017 outbreak, we saw 2 lineages develop with H7N9. And so when you talk about H7N9 in general, you can't assume it's one virus, because each of these branches on this tree is an H7N9 virus. It just shows the difference in the genetic sequence of the hemagglutinin gene. There are also now 2 lineages in China. One is the, and I probably won't pronounce it properly, the Yangtze River Delta lineage, which is at the top, and then the Pearl River Delta lineage, which is at the bottom. The risk assessment tool used the Yangtze River Delta lineage for its assessment. Oh, and I should also mention that the researchers are working on candidate vaccine strains for all of the lineages now, not just the general vaccine strain that's listed on this slide at the very bottom. As you can note, the current candidate vaccine strain for H7N9 is in the Pearl River Delta lineage, but a lot of the current strains we're seeing are in the Yangtze River Delta lineage, and they need, due to mutations and differences in these lineages, they do need candidate vaccine strains to cover both of them. These strains in these lineages are not in the United States. The United States lineage is not listed here just for ease of slides, right? I don't even think it would fit. It's very difficult to show the phylogenetics of all of the different strains, but it's a completely different branch on this map, just to give you an idea. And so to talk about the United States, there was, I'm just going to pick one, although if you go to the USDA APHIS website, you can find information about other states that have had outbreaks of H7N9. In Tennessee, there was an outbreak in March. And again, this is genetically distinct from the Chinese strains that we're seeing. It was a highly pathogenic strain from the wild bird lineage in North America. However, as compared to the cases of human infection with H7N9, historically here in the United States, we've only had 10 to 15 cases of avian influenza, and they've all been people who have direct contact with live birds. So there were other outbreaks in 2014 and 15 with the H5 version of high pathogenic avian influenza, which is the one up until a few years ago that when we talked about avian influenza, it was all H5N1, and now nobody's talking about H5N1 anymore. They're all talking H7N9, but H5N1 is also prevalent, and we did see it here in the United States, primarily in migratory bird paths in the south. And again, the USDA is a really great resource because it is mainly an avian infection. Their information is going to be a bit more detailed than the CDC website, although the CDC website is a really great resource for public health professionals. But the USDA will give more information on the specific outbreaks, their actions, and follow-up plans, because again, most of the planning and response to these outbreaks is to close the farms and to cull the population. So what do we know about avian influenza? So we know we have a lot of clinical strains, and there are a lot of scientists who are working in the avian influenza research field. But the avian influenza research field, I felt, as a biosafety officer naturally and a former researcher, I couldn't not discuss this topic because it has been a real regulatory shifter and shape and develop in a process developing item for us since 2013. So what am I talking about? So back in 2012, a few different flu researchers submitted papers for publication discussing how they created the H5N1 avian influenza virus. They studied it and in one case created some mutations, and another one they let it mutate naturally that allowed it to transmit between mammals by the respiratory route. And as we've discussed here today, that's not common, right? It needs mutations, and it needs to have a different binding potential to do so. So these papers created a lot of fuss around the topic of dual-use research of concern. And it actually caused a moratorium on all avian influenza research and what they call gain-of-function research. The gain-of-function research is where they, the researchers in the lab can take each of those 8 genes from avian influenza, they can mutate them, and then they put them into a cell, and the cell can produce the influenza virus. It's a pretty straightforward technique that's performed at biosafety level 3 facilities with certain enhancements for protection of the people and also the environment. It's highly regulated, but due to the concerns with whether or not ethically we should be doing this kind of research, it was shut down. Today is not a time to discuss the ethics, but I did want to point out that the National Academies of Science did a very in-depth study on dual-use research. We now have 2 policies in the United States covering review of research that has dual-use potential, and working with highly pathogenic avian influenza is one of the agents that must be reviewed for dual-use research. And so the reason I point this out is that a lot of these strains are now showing mutations that are moving the H7N9 from the low pathogenicity scale to the high pathogenicity scale. which, as I mentioned, makes it a select agent. It also makes researchers subject to the dual-use research of concern policies and specific approvals by CDC and USDA to do the experiments. And so I think that the folks that are already publishing data, and there's been a lot of papers on H7N9 already. There are papers looking at vaccine candidate studies. There's a lot out there, and these are all coming from our big flu labs that have been able to continue doing this kind of work. And there are other labs, some here in the tri-state area, that do similar work or can do very simple infection models to help study this as well. But once this change was noted from the low to the high pathogenicity scale, it does make sharing strains and doing research on these agents a little bit more cumbersome, although it is ongoing and still very important. And seeing these mutations happen, I think, also explains why researchers are doing and trying to anticipate some of the mutations that occur, so that way they have a better idea of how to make a vaccine and also how to develop better antiviral therapies. In addition to some of the research studies that we can find, the strains that are coming out of China already have mutations that are known based on the what they called the gain of function research that they were able to identify that confer greater ability to transmit via the respiratory route between mammals. Some of them are known to have the mutation that confers the high pathogenicity. They know where that one of those mutation lies. And they also have the mutations for a lot of drug resistance, which is common. We also have the epidemiological data that is showing the severity of symptoms, the location, and people who are becoming infected with these viruses. So I'm going to switch gears now to talk about the influenza risk assessment tool on the CDC website. The CDC makes it pretty clear, though, multiple times, sort of funny when you read it, that this is an evaluation tool only. It does not predict pandemics. And so just because they've ranked this virus as a high potential for causing a pandemic, it does not mean it will cause a pandemic. It's just that they've ranked them and thought about the viruses in different scenarios, and they come up with a rating, and it shows the potential for that virus to cause a pandemic. So they have two scenarios that we'll discuss because they take the evaluation criteria and weight them differently based on each of the scenarios. One of the scenarios is emergence of these viruses and having them emerge in the public and having them cause disease, and the other is if they were to emerge and cause disease, what would that impact be on both the public health population, nursing staff, hospitals, but also just globally. The evaluation criteria is broken up into three categories. The first is the properties of the virus. So this is going to look at genetics and that kind of thing, attributes of the population, and that would be more of the human side of it, and then the ecology and epidemiology of the virus. So breaking it down a little bit more on the properties of the virus, this is where you look at all the different mutations that the genome has, and if it has a certain mutation for drug resistance, or a certain mutation that causes the transmission between mammals, this would be where they would look at that. Also the receptor binding potential, which does it bind to the 2,3 or does it bind to 2,6? That's going to be really important. They use the lab animal studies that they have on transmission to look at the different viruses and the transmissibility between animals. Typically, they study direct contact and also aerosol challenge. And then finally, they look at any susceptibility or drug resistance. In the attributes of the population, they're going to consider any existing immunity. Our vaccines that circulate right now do not contain H7N9, but when they look at across the board at the different viruses that they're going to consider, because you'll see there's more than just H7N9 listed, they're going to look at the potential immunity in the population to that virus for the help protect them. They look in general at what is known with the disease severity and how likely it is to spread between persons. And then also, how closely is the relationship in terms of that hemagglutinin and neuraminidase antigen on the outside of the virus particle to the vaccine candidate virus? So if you think about the phylogenetic tree I showed you a few slides ago, if you had a virus that was all the way at the top compared to the vaccine strain, which is all the way at the bottom, their proteins on the outside are going to be very different. And so the more different they are, the less likely that vaccine is going to confer any protection. So that's why they're looking at candidate vaccine strains across the different lineages and in different families to ensure that they have vaccine candidates with very close antigenic relationships. And then finally, they're going to look at the ecology and epidemiology. This is where they think about, where is this infection found naturally across the globe? So people in China, for instance, may have more natural immunity than somebody in the United States, just because we don't see these viruses here. And so somebody there may have come across a low pathogenicity avian influenza, maybe had mild symptoms, and now have immunities to a certain type. They also look at the ability of the virus to cause infection in humans and animals and evaluate that. So the first scenario is emergence. So what is the risk that this novel virus has the potential for sustained human-to-human transmission? And so I'm not going to read over the list, but you can see this is how the CDC ranks the previous 10 evaluation categories. And then based on the number that's scored with these categories, we'll give them a number at the end, and I'll show you a chart in a second. And so here, the most important concerns are things like human infections, transmission, receptor binding, immunity, etc. Whereas in the public health impact, this is the scenario for the ability for the virus to sustain human-to-human transmission. So it's more than just the 1 to 2 people. It's if I have H7N9 infection, what is the likelihood that I could spread it to many people? And then what is that impact on public health? So you'll see that receptor binding is more at the bottom of this category, whereas here we're looking again at immunity, but we're also looking at disease severity and pathogenesis. If a lot of people are ending up in the hospital because they need health assistance to breathe, that's going to put a huge impact on the public health infrastructure of our country. They also look at antiviral treatment and susceptibility. So that way, if this is an outbreak, will the antiviral medicines that we have available be sufficient, and will they work? And so this is the different ranking here. So this is the results that are published on the CDC website. This is done earlier in 2017. So I'm sure they're updating it as different viruses are evaluated and different things are seen. And so as you can see here, the H7N9 viruses are listed right at the top. One of the 2017 lineages, which is the Yangtze lineage, is right there at the top with an emergence score of 6.5 and an impact score of 7.5. And the 2013 virus isolate is a little lower. But again, both of these viruses were of the low pathogenicity. And so they most likely the CDC may release information as they assess the new high pathogenicity strains. The H5N1, so there's a lot of different clades, which is another way of saying, separations, families. One is at E, and then a couple are actually further down the list now with H5. But then you see a lot of other avian influenza viruses ranked here. So now I'm going to transition away, but if you want more information, I should probably just say, the CDC website, and if you Google risk assessment tool, you'll come up with the website, and it gives you a lot of information. They have a lot of different charts and graphs, so you can understand how they're looking at the risk assessment of these viruses. And as you can see, they're basing it on a very specific virus. They're not basing it on the entire virus family of let's say H7N9. And so now I'm going to switch over to symptoms, transmissibility, and worker protection. So the symptoms, a lot of times they're seeing, they can see influenza-like symptoms, which are cough, runny nose, fever. They're also seeing a lot of pneumonia cases if somebody is infected with H7N9. But the most important thing is the likelihood in the United States is low, but if somebody has been working with live birds and they exhibit these symptoms, then you may want to consider that it could be something like H7N9. But again, the occurrence here in the United States is so low that this is really something that's more important if somebody's been traveling across various countries and has been involved with any kind of live poultry markets or things like that. It kind of brings us back to the Ebola epidemic where people were being screened, have you traveled, what kind of contact have you had, things like that. Transmission of these viruses, if they are able to transmit between person to person, it's the same as normal influenza. There's droplet spread up to 6 feet in distance, so you want to use proper respiratory protection, but also, just the general hygiene, coughing or sneezing into your elbow, making sure you use a Kleenex and throwing it away, washing your hands, and things like that. Transmission can occur 1 day before symptoms, 5 to 7 days after symptoms start, and so it is very similar to what we would consider regular influenza. And so when we're thinking about preventing H7N9, I work with a lot of researchers, and we joke because we do some H7N9 work, and there's a quarantine policy from the USDA, so they call it a quarantine policy. We joke and call it the don't touch the birds policy, because if I say quarantine policy, they forget what I say. And so that policy says that if you're doing research or working with H7N9, you cannot have any contact with any birds during that time and up to 7 days after you've stopped your research. And so we have some researchers who cannot do the work or cannot go into the laboratory because they have a pet bird. And so it's really important, but that is really the message. The message is do not have contact with birds. It's hard. I live near a really nice park with a pond, and my kid loves ducks, but I know too much now, and it's like, okay, stay away. You can feed the duck, but stay away. So that's what's really important. And also, this gentleman here says, I'm from the CDC, he's holding a chicken. That might be the case for a lot of folks. We may have farmers, people have chickens in their backyards now, and so it is always possible for those animals to be harboring some version of an avian influenza virus. And so if we go and touch those animals and then touch our faces, we can easily spread them. It's also spread through their excrement, which can be aerosolized within their little housing areas. And we don't know they're sick because they're reservoirs for these viruses. If you are in an area like a poultry market or a farm or even someone's backyard, and the animal looks to be sick, definitely don't touch it. But even just the animal moving around, shaking its wings, scratching its head, all of that can aerosolize the virus and cause us to breathe it in. So that's the major route. And this is also why the USDA has this policy, so that way if, as researchers or people working in your labs or even in public health labs, if you isolate one of these viruses, you probably have to follow the same quarantine policy. So it's so that way we do not spread that virus out into the population. So they have very strict guidelines for lab waste and water and all of that. In addition, you would think that the annual flu shot would not be recommended, but it is recommended. Why? Because again, we don't want to be infected with a human flu virus and then accidentally be infected with an avian flu virus and then cause these viruses to recombine and make a new one. So if you are working with or could be working with these kind of viruses or have contact, it is required by USDA to have the annual flu shot. Most clinicians are required to have this anyways to help prevent transmission in public health settings and in hospital settings and doctor's offices. I know this year's is thought to potentially not have a lot of efficacy, but it's still important nonetheless. As with any potential respiratory acquired infection, it is really important for all clinicians to be wearing N95 respirators if they're working with somebody who has the potential for a respiratory infection. A lot of times, flu infection is so common, especially in the winter, that we see clinicians not donning the respiratory protection. Somebody may have a cold. You may not want the person, your patient, to think differently if you're donning this respiratory protection. But if you think that people are sick or you're seeing a lot of spikes, it is the responsibility of healthcare workers to help prevent the spread of these things and also to keep themselves safe. And to keep yourself safe, the best thing to do is to wear a properly fitted N95 respirator. Cough etiquette, putting a surgical mask on the patient so that way they can't cough everywhere, that kind of thing. And also screening patients. So if you're a dentist, you don't want somebody coming in if they have a cold or the flu, because you're going to be aerosolizing everything in their mouth while you're working on their teeth. So it's important across the board to screen patients. People who are sick, don't like to stay home, but it's really important for them to stay home and not spread. Also in healthcare, the CDC website has a lot of information about implementing environmental measures, different cleaning processes, although flu viruses are very susceptible to all disinfectants and are very easily killed. But also using engineering controls and separations, negative pressure rooms, if you know somebody has an infection with these viruses. And again, monitoring and managing any ill healthcare worker. Typically, standard flu is droplet precautions, and also, if possible, avoid any aerosol generating procedures unless you cannot avoid them and they're really necessary for that patient. The CDC website also has a lot of information on pandemic planning. The most important thing that with avian influenza, they still do talk about making sure you have a vaccination program in place and making sure you have adequate supply of vaccination. Again, just to make sure that people decrease the likelihood of being infected with a human influenza strain to prevent any kind of cross-mixing of these viruses. It's also very important to have adequate antiviral medications on hand, and as opposite to the normal human viruses that we have, where we have a lot of different options of antivirals, we cannot use the neuraminidase inhibitors for avian influenza. And then the last thing is maintaining respiratory protection devices. So as we saw with Ebola, after a while it became very difficult to get more N95s. The suppliers were running behind on manufacturing them, and because they're disposable, people were wearing N95s more than usual, and we were shipping them overseas. And so maintaining an adequate supply, but making sure that they're in good shape and not damaged or wet, it will help with the pandemic planning as well. And so like I've mentioned, these viruses, the avian influenza viruses, and specifically H7N9 as well, are typically resistant to amantadine and romantadine. The two the antiviral therapies that can be used are the Tamiflu and Relenza, which are the trade names, and those are the ones that are really the go-to. Unlike with normal flu infection, where doctors may not recommend an antiviral medication because if you don't take it soon enough, it's not going to be effective, persons with potential H7N9 infection are pretty much put on an antiviral immediately. They do this for even people who are exposed to H7N9 because the symptoms are so severe and mortality is very high. And so they will put you on the antivirals to help avoid infection or to lessen it as much as they can. So I received one question about the medication, so I hope that what I've just mentioned was clear. If not, let me know and I can add some clarification. And we had another question about if I'm aware of procedures used at poultry processing centers to guard against avian viruses. I don't specifically know, other than I know that they're trying to do more like cold chain transport and storage and improve those processes, disinfection use. I think the United States, of course, we have very stringent food safety requirements. And once you cook poultry, I should mention, the risk goes away. So if you cook the chicken and the eggs all the way through, influenza viruses are very easy to kill. And so it's not something you have to worry about if you're eating cooked food. It's really more of making sure that you're disinfecting areas and cleaning things appropriately. And so I know with the reading that I've been doing in China, they've been trying to step up their cleaning and disinfection and doing even larger scale farming and centralized slaughtering, so that way they don't send live birds to many different markets, because then when you have multiple birds and they're all alive in these markets, that's when they're sharing the viruses. So if those kinds of practices are put into place, hopefully that will lessen the issue. All right, and then to summarize, H7N9 does possess a very high pandemic potential due to the additional mutations that have allowed some of these strains to become highly pathogenic, but also due to our lack of immunity. But to date, just to remind you, no sustained human-to-human transmission has occurred with these viruses, although there have been some person-to-person transmission documented. And again, just as another take-home message, our strains here in the United States are different from the strains that are being seen in China. Although, with global travel, anything I guess is possible if somebody were to be sick. But again, I think the likelihood of that could be very low. So one of the questions I just received is, that I mentioned that individuals getting sick are quickly developing pneumonia. Is this different than the symptoms from other avian and flu viruses? Well, I think the issue there is that there's not a lot of data to go from with avian influenza infection. I think these outbreaks that we've seen in China have the highest amount of people infected, and prior to 2013, there were no infections with H7N9. I think in general, avian influenza viruses, when a human acquires that infection, the symptoms are always more severe, and the potential for severe respiratory effects from influenza are always prevalent, or especially in more high-risk populations. But they're seeing these viruses are causing severe pneumonia. But again, there's not a lot of data to go on comparing across the board. This is probably the most data we have. And so there's a few other questions coming in. Oh, thank you. So basically, to conclude, I would say as public health professionals, keep abreast of the CDC website for more information, also the USDA website, especially as these strains continue to evolve. I'm sure the CDC will put out additional risk assessment tools, but the planning items and things are really no different than what you probably have already been dealing with and preparing for other flu pandemics. And thank you so much for your attention. I'm happy to take any additional questions if you have them.